Welcome, students, to one of the most popular segments of the course, which is on ancestry. So we're moving again through the biological profile, and then we're going to have a final lab practicum where you're going to assemble it in a number of different components, right? Because we've already done sex determination, age at death. We're moving on to ancestry, living stature, and even pathology if we can. So this one lecture series is broken up into two parts. In this one, I'm going to talk about the concept of race for the most part because it's important. And hopefully you'll understand a little why I've gone to a great extent to explain it this way. As we mentioned, we are in the biological profile. We've looked at sex determination. Remember, sex, not gender. We've looked at chronological age. What's the best for subadult and what's the best for adult remains. Be thinking about this when we get to your final lab practicum. Ancestry is what we're going to work on today. Can we actually look at remains and figure out like a general category from where their heritage, which continental group that individual or that decedent comes from, right? Because we're looking at skeletal remains and it's somebody that's likely a victim of foul play, homicide, suicide, for instance, missing persons, for instance. Very good examples. And when we have descriptions, those descriptions usually include something like ancestry. But you may also see things like race that's used, even on recent forms. I hope it's falling by the wayside, and you'll see the way I'm explaining it. First we have to understand what the concept of race is anyway, and what is actually ancestry. And so, again, this part is the concept of race, and the next part is ancestry. Then we'll move on to things like living stature, which, by the way, you do need to know ancestry, have a good idea of ancestry to determine living stature, and that's why it's pretty important, because we know that people, in terms of how tall they are, like their height, their living height, that there's variation among people that are African American, for instance, in comparison to people who have Euro-American ancestry. So this is important to understand if you want to make a stature estimation. We'll get there, and if we have time for pathology, it will be integrated. So what about race? What does race mean? Number one, well, oftentimes it's used presently as a social construct that we pretty much created. Race was a biological term. Many people use this term late and early 20th century, and some of them would describe it in terms of a new discovery, like a new race of beetle, a beetle that an entomologist discovered and had never even recorded before, and then they would use race to kind of describe it. But then it got picked up by a number of different individuals near the end of the Enlightenment, and it was used to mean different groups of people, in terms specifically humans, and then we can divide us up into some of these groups that are created, and yes, some of them coincide with things like latitude, some of them coincide with things like geography in terms of different continents. But the question always remains, if this is a social construct, which I can kind of show you clearly that there are components of that, is there anything about the concept of race that has any value for biological variation? Now, we're going to look at some minute biological variation in the skeleton, specifically the skull, right, in the cranium. And those very small details may actually help us in determining something like ancestry. But again, these ideas are usually termed something like race. And so let's explore that. What is race in terms of, it being this construct, does it have any biological variation? If it did, then it wouldn't quite be a social construct, if you know what I'm saying. What you're really looking at is human biological variation. Now, we know what it makes to be us to be human. And, if you want to know more about this, you would take like an ASM 104 class, Bones, Stones, and Human Evolution, where we talk about the fossil record through the changes, particularly in the cranium and the skull, that we notice that eventually will evolve into us. And there are a few components and signatures that demonstrate that an organism is absolutely us. And one way to do this is in an area on your brow ridge known as the superciliary arch. Now, there are a few other components, but this is on the supraorbital ridge, right? And on the frontal. And, also termed the brow ridge, and we know that males actually have larger ones, right? So you're with me? There's a slight disintegration in the brow ridge, and this is known as superciliary arch, and you have one on each one of the superior aspect of your eye orbits. And this makes you human. And then we can connect all these other features to other human-like things like Neanderthals, as well as Homo erectus, and then even other creatures that predate, those creatures that are in the genus Homo, like Australopithecus. And some of these that date to, goodness, I think some of the oldest creatures that are intermediary between apes and us date to about 5 to 8 million years old. Some of them 7 million years old. And DNA also tells us that we last shared a common ancestry with chimpanzees and potentially other African apes 5 to 8 million years ago. And so this is what's known as common ancestry, that we can look at traits that we actually observe, right? And we can see it in the skeleton, but it can be other traits that we observe in DNA. It can be sometimes morphology, which is the shape of organisms, like size and shape, as well as we can look at things like color. I mean, when you look at, things like birds and lizards with different colors, you would assume that they would have similar genes, and they have to exhibit those traits because they have a closer common ancestry through time. And the key assumption, of course, with common ancestry is that all carbon-based life forms on Earth are biologically related in some way. It's a pretty good assumption in evolutionary biology. That would mean that life evolves, and we've talked about evolution. That was some of the first part of this class. And the key is time, right? And evolutionary time. We use geology as a way to determine evolutionary time, but it's only a proxy, right? And when you look at time in an individual or organism in space, then that would also mean that determines whether you're talking about ancestors or descendants. For instance, if you look at me, I was a descendant of my parents, and then once I had a child, now I'm an ancestor, right? So typically, time as well as creating offspring, then we get to see little slices and components of evolutionary history. And of course, the hardest things to determine in science is how and why is always and when, of course, because we're talking about time. Those are the pressing questions. So here's another way to kind of look at it, so you can kind of wrap your head around common ancestry with phenotypes. Those are some of the traits that we can see. And we assume that there's a strong genetic component for an organism to display similar traits. Now, it doesn't have to be identical, as well as in terms of their DNA composition, but we have to have something, right, that will designate that it is a physical trait that we can see. Now, there are other components that the environment plays that will also shape what we view and record within an organism. For instance, your stature, how tall you are, your height, is determined by a number of genes plus your environment, how many nutrients you got in your environment. Because if you don't, then you have growth deficiency and you will never reach your genetic potential for height. So we know that the environment also plays a role in the shaping of and changes and differences between and within organisms. But if we look at the organisms that are on display here, human, horse, dolphin, and a bat, what's a way that we can connect all four of these organisms? If you said mammals, you're right. Each one of these is a mammal. And there are a few distinctions for what a mammal is. There's a few traits that can be easily and readily used. Like, for instance, 5 digits on the terminal appendages, right? If you look at your hands and your feet, you have 5 on each one of them. You have a thumb and 4 fingers. You have a big toe and you have four little toes. Among all mammals, with notable few exceptions, all of them have 5 digits. If you look at, for instance, the bat, the wing of a bat, a mammal that is adapted for flight. If you kind of like spread their wings and extend them out, and you can actually see within the webbing of their skin, there are 5 digits. If you look at a dolphin or other orcas and whales, those are pinnipeds. These are mammals that are adapted to water and swimming. Like the fin of a dolphin, for instance, on the end of each one of their appendages, there are 5 digits. If you look at a human, 1, 2, 3, 4, 5, that's you. As a matter of fact, among mammals, this is a trait that's been fixed by natural selection probably 50 million years ago. Maybe even a little older, 60 million. And so think about it. If I see another creature that has 5 digits on the end of their terminal appendages, I could go 1, 2, 3, 4, 5. I think that's a mammal. You got a pretty good bet in terms of, put some money down that you're looking at a mammal, a creature that is a mammal. Now, what are some of the exceptions? There's a spider monkey. They typically have a thumb that's a little more descended and smaller. You have a panda, which actually has on their hands has 6 digits. They have another bone called a sesamoid that is kind of like another bone that extends into pretty much another thumb or finger, and they've actually evolved this because it helps them hold bamboo easier. So again, you'll see that there are notable exceptions, but for the most part, the 5 digits is very good and useful. What about the horse, right? That's the one I haven't talked about yet. Well, horses are really strange if you ask me, because we do have horses that, matter of fact, horses evolved on both continents, in the Americas as well as the Old World, parts of Africa, Europe, and Asia. And then they went extinct in the Americas, millions of years ago, and then they were reintroduced by Europeans. But if you look at modern horse and compare them to fossil horses have 5 toes, then they have 4, 3, 2. Eventually, we see a nice progression of the loss of each toe over time. And then, of course, we're curious what it means. But modern domesticated horses, they only have 1 finger. And they have an exterior hoof that also evolved, made of keratin, really hard protein that's in our nails and our hair, and is made of other things like horns. And that means a horse is literally just walking on their fingertips. That's why, to an anatomist, horses always look strange. I mean, super cool, but I always just picture them walking on each one of their fingers. And this is kind of what we just did. We looked at polymorphism. We looked at, okay, this 5 configuration is pretty good in mammals, but it is still polymorphic, meaning there are some organisms that display differences, like the horse, as well as the panda and a spider monkey. And there are probably a few other examples. But what we need to do is have at least some type of variation in the genes in order to see those differences. So polymorphism would be when two or more phenotypes are present. Those are phenotypes, things that you can actually see, record, color, for instance, size, number of digits. When you look at DNA, you only need to have one single nucleotide polymorphism. That is one single base pair difference in your DNA to suggest that there are differences at that one point or locus on a gene. And so you can look at these two chicks in the picture at the bottom. If we look at in their cells and then we type their DNA, we can see that they have 3 spots that are different. Now, where one has a C or a G and A, and T and A and a G, vice versa. And so this would mean that those sites are polymorphic, single nucleotide polymorphism. And we've got 3 of them in this case between those 2 individuals. And, we're not sure if that DNA actually creates a difference that we see in their features, but we know it's a difference at the genetic level. And if you look at humans, because that's where we're transitioning, if you look at humans typically show 28% polymorphic loci. So in our DNA, only 28% of it has differences in its genome. And just get a hold of our genome, our nuclear genome is typically billions of base pairs. And that would mean that 72% of us in our DNA are the same. And so when you try to figure out differences between organisms, you don't typically go to the 72% because that usually means that it's all going to be the same. You can't find differences. So you have to go to the 28% polymorphic loci in the DNA, and that will tell you about where some of those differences, as well as locating them. Now, at the same time, think about it. If we're talking a lot about, we're so different, if we're 72% the same, doesn't that mean we're not really highlighting how similar we are? A few interesting aspects of some parts of the genome that are consistent and then the same can tell us about time. So when we look at, polymorphic parts of the genome, it tells us about differences. But then if we look at, for instance, in this chart here, we look at a gene called cytochrome C. It's actually like hemoglobin. And so it would be effectively, we can look at, a number of organisms where hemoglobin has evolved, and you can see a bunch of mammals on here. And you notice that in this graph, it has 2 axes, X and Y. The x-axis is looking at millions of years, and the y-axis is the number of substitutions. Those are the number of changes that we see between organisms. And you see a nice bisecting line, almost pretty much a 45-degree angle bisecting a 90-degree angle, which means typically a 1-to-1 ratio. And you can see there's a lot of organisms compared here, like a human and kangaroo, human cow, dog cow, rabbit, rodent, pig cow, goat, sheep, goat, horse, donkey, what have you. And this is what's known as a molecular clock, that for genes that don't change very quickly, those that are conserved, that the mutation rate is relatively consistent over geologic time. So you can look at, for instance, the organisms that are compared with humans. On this chart, it shows a human and a kangaroo, a human and a cow, and a human and a rodent. Now, that would mean that we are most closely related as humans to rodents, and that's at about 80 million years ago. It was the last time we shared a common ancestry with them. And that's probably true because primates, remember, is what we evolved from. And we think that primates evolved from small rodents like tree shrews, colugos, flying squirrels, um, and the like. And then you can see a cow is also a mammal, and a kangaroo is a different type of mammal, because a cow is a placental mammal and a kangaroo is a marsupial, where the baby actually lives in the pouch, right? It's born kind of prematurely, but then is nurtured in a pouch. They're more primitive than placental mammals like a cow, you and me, and a rodent. So that makes sense. So when we look at the information that were very similar, then we can actually look at when we last shared the same lineages. And this is how we introduce coalescent theory, right? We can look at and sample a bunch of different organisms within a lineage, and then we can trace genealogies. You can see that some individuals they don't reproduce, and of course, when we sample populations in science and in research, we're not going to sample everybody. It's just impossible. You can't, do all the research. You can't sample everybody. And so there's going to be bits and pieces missing here and there. And typically what we do is we use this idea of coalescent theory, and then it will transform into some of these types of coalescent tree of the sampled lineage, which you see on the far right. So we'll think about coalescent theory, and then it'll transform into some of these more sophisticated trees that I will show you. We can look at this neutral variation for conserved genes in a few ways. We can look at it between organisms, like you saw before, like a human and a cow, a human and a rodent. And then we can actually do it within species. So we can actually test, everybody at Phoenix College, part of their DNA that's conserved, and then we can create what's known as a coalescent tree. That's coalescent theory here, where DNA tells us about time. Your DNA tells us about all the adaptations and things that have accumulated over time. And then we can look at those changes between individuals, and then we can go back generations. And we've done this with other types of DNA, like mitochondrial DNA, which only is inherited in the female line. The male line is Y chromosome, right? That's pretty consistent. And so what we've talked about is tracing back to the mitochondrial Eve, because that would be, right, the first woman, and people have talked about the Y chromosome Adam. So using our ideas of coalescent theory, we have a number of different, coalescent trees here. And as you can see, we have two of them, and one is mitochondria and the other is Y chromosome, right? Maternal and paternal lineages. So these are specific pieces of DNA that are conserved, that are fairly neutral in terms of their adaptation rate, evolutionary rate over time. And they're only found in one side of the family tree, your mother's side or your father's side. It makes it a little easier to do this work, because then you don't have to deal with some of the complications of DNA inside the nuclear genome. And these were done around, the turn of the millennium. And you can see that with our ideas about coalescent theory, they've kind of been transformed into these, coalescent trees. And you can look at the base of the trees. The longer the lines, you typically mean the older the lineage, right? Because we're looking at things like time. And you can see that both with mitochondrial DNA and Y chromosome, the oldest and longest lineages are all in Africa. And that typically means that human evolution into our species, Homo sapiens, likely occurred in Africa. And then you can also see this red dot on both of them, red star, more so, that actually represents the common ancestry of every other non-African person. So you can see that there was like some type of probably exodus from Africa and then a radiation after humans left Africa. So there's much greater genetic diversity in Africa. And then as you move further away from Africa, then genetic diversity goes down. And this pattern is the same for both mitochondrial DNA and Y chromosome. But you also notice that after that red star, there is an African population that is on the other side, right, of that split. And why is that? It's a good question. Well, I'll tell the answer. You may be able to guess, but pause it now if you want to guess. But what we think is that's a back migration to Africa, that those are populations that may have left Africa, and then we're like, this sucks. So they went back to Africa, and we know that there are some populations like the Yoruba who are those that live in parts of Central Africa, that Central and West Africa, that they have stories of back migration. What these researchers also did was use those time signatures to look at the historical pattern of migration throughout the entire, the whole planet. And so what you notice is that, for the most part, right, human evolution occurs in Africa, and then and this occurs around 200 to 300,000 years ago. We have other fossils that verify this in several regions. And then humans leave Africa at about 100,000 years, then start to populate parts of Europe and the Asian continent 40 and 50,000 years. Humans may have made it to Australia as old as 70,000. This chart is actually almost 20 years old now, but it's still pretty accurate. However, we've made new discoveries in Australia that are 20,000 years older than what DNA actually predicted, that there probably would be older populations in Australia. That was neat. Archaeologists went out and verified that, and doing all sorts of cutting-edge research, finding things that are 60, 70,000 years old. And then we think that, the Americas were populated with DNA 35,000 years ago is the oldest date we get, though most of the sites that are that old are more securely dated to 15 to 20,000 years. And when we look at some of our, association trees, coalescent trees, it looks like Native American people, or people that are native to the Americas, that they are most closely related or cluster with people from the Asian continent. And so we think that there was a migration from places like Siberia into the Americas via Bering Land Bridge, or glaciers, ice that they could walk on to get to the Americas from Asia. You also have to understand that more water was taken up by the polar ice caps at this time. So the water levels were lower. People could have made it through maybe a coastal route as well. And some people have talked about an ice-free corridor into the Americas. There was a little criticism with just using mitochondrial DNA and Y chromosome, but then we certainly were able to integrate the nuclear DNA, which has many more data points in terms of how much material. Remember that you're talking about billions of base pairs. When you get to mitochondria and Y chromosome, you might be dealing with things like 10,000 or so. And this showed the same pattern in nuclear DNA as in mitochondria and Y chromosome. The oldest lineages are in Africa, and then there's a periodic exodus away from Africa, as well as reduced diversity as you move away from Africa. You can notice the Asian populations, they are closely related to those that are in parts of Southeast Asia as well as the Americas. So that kind of agrees with our previous research. So I talked about how, what are we trying to do from the skeleton we're looking at skulls and we're thinking about this concept of race, we should have a good idea of what it means to be human and what the concept of race and how does it at all or some value, does it have some value as a way to describe human biological variation? Well, I've shown you that with genetics and neutral variation, it tells us about time, right? And migration. Now, can it really, can it have any other importance other than that? Well, I can show you when we look at the DNA of specific populations, right? Let's try to think about if we were to splice up each one of these continental groups and then compare them to each other and their differences, what would that tell us? So that would mean, how are the differences among us on particular continental groups, for instance? How is it distributed? Well, overall, the genetic differences between humans on continental groups is very low, surprisingly low. That's because we're fairly young organisms. We haven't had a lot of time to evolve on this earth and then evolve away from, subpopulations. And there is some of that variation, but goodness, only like 5 to 10% of that variation occurs between groups. So think about that. We can create all these groups in our head that we've constructed socially and culturally. And then when we do that, we compare those groups, like from Africa, from the Americas, from Asia, from Europe, and there's only like a 5 to 10% difference. But then if we go to Africa, if we go to the Americas, we go to Europe, and we start sampling DNA, wow, there's actually a big difference between the localized groups. 90 to 94% of that variation occurs within the groups. Wow. So what does that mean for, the race construct in terms of having biological significance? What it typically means is that, yes, each region will have, particular region of humans, geographically, continentally, by latitude, because we know skin color, skin pigmentation is something that correlates with latitude as well as solar radiation. And there's going to be a limited number of features, and that's kind of what we're doing. We're using a limited number of features on the skeleton to try to get at what we're going to use ancestry and what others may think of is race. But I'll show you, and I hope you've seen that race is a social construct, although it's still important to talk about, because in the concepts of critical race theory and racism, people who have power, authority, and feel superior over others because of those features, certainly that's important. But race typically, as a term, doesn't really have any biological significance.